The title of the presentation is called Regulation of MMP Activity by Nci Molecules. So starting with the abstract, matrix metalloproteinases are a family of enzymes primarily responsible for the remodeling and degradation of the extracellular matrix. The overexpression of MMPs is associated with various types of cancer since the rapid breakdown of the ECM removes physical barriers in tumors, facilitating growth and metastasis. Therefore, the discovery of MMP inhibitors as anti-cancer agents would be therapeutically beneficial. Additionally, underexpression of enzyme activity also disturbs their normal regulation, making the search for activators a target of interest as well. Our lab previously performed computational binding of National Cancer Institute diversity set 2 ligands to MMP9. Here, we studied the effects of 4 Nci molecules on MMP1, 3, 9, 10, and 12 by fluorometric assay. Upon assay completion, initial rates were calculated and binding constants Ki and Kd were obtained to determine the overall effects of enzyme activity. MMPs are a family of proteolytic enzymes that are lowly expressed and tightly regulated in normal conditions. When they're overexpressed, they contribute to pathologies, most notably different types of cancers, such as breast, prostate, lung, colon, gastric, and pancreatic. For this reason, inhibitors of MMPs are sought as anti-cancer agents. Underexpression of certain MMPs has also been linked to cancers. Therefore, activators are also being investigated. MMPI, such as sulfonamide and hydroxamic acid derivatives, have failed clinical trials due to their broad spectrum inhibition. These nonspecific molecules are disadvantaged because they may unnecessarily inhibit non-target MMPs or other metalloenzymes. 23 MMPs exist in humans. They're divided into 6 subgroups based on structure and substrate specificity. And so these are collagenases, gelatinases, stromolysins, matrolysins, membrane type MMPs, and non-classified MMPs. Our lab previously determined a computationally screened binding affinity for L1 against MMP9. Here we investigate the interactions of L1 and 3 additional molecules against other MMPs through enzyme kinetic assays. Binding affinities are a measure of the interaction strength between a receptor and its ligand, with a lower binding affinity indicating that more energy is required to break the interaction, meaning that it's more stable. As you can see, the structures of the Nci molecules, L1, I1, I2, and I3. So the objective is to observe the effect of Nci molecules on the enzyme activity of various MMPs. So as for the results, Figure 1 shows the binding of I3 to MMP1, and Figure 2 shows the binding of I3 to MMP9. And seen here are the predicted pharmacophores of the docked molecule in the active site. I3 had a lower docking score with MMP9, indicating a stronger interaction to this enzyme compared to MMP1. One second. Sorry, I'm having trouble. That's weird. Okay. So for Figure 3, it shows the initial rates of MMP1 versus concentration of I3. And for Figure 4, it depicts the initial rates of MMP9 versus concentration of I3. So here we see the effect that increasing concentration of I3 had on the enzymes, which was inhibition. And although I3 showed stronger binding towards MMP9, as previously seen, it turned out to inhibit MMP1 far greater, as seen by the significantly smaller Ki. And as you can see, a smaller Ki equals better inhibition. The results for L1. So Figure 5 shows the initial rates of MMP1 versus concentration L1. And Figure 6 shows the initial rates of MMP9 versus the concentration of L1. And here we see the effect that increasing concentration of L1 had on the enzymes. Our lab had previously screened this molecule against MMP9, but now we see a greater activation against MMP1. So smaller KD means better activation, and L1 had a smaller KD for MMP1 than it did on MMP9. And I've had forgotten to mention the materials that we use. So We use MMP 1, 3, 9, and 10, and 12, and fluorogenic Omni-Red substrate. Omni-MMP red substrate were purchased from Enzo Life Sciences. The small molecules being tested were obtained from the National Cancer Institute Diversity Set 2. The buffer used was 50 millimolar tris HCl and 5 millimolar calcium chloride with a pH of 7.5. Assay wells contained zero point 0.5 micromolar of substrate and zero point 0.9 units of enzyme, with final concentrations of Nci molecules ranging from 0 to 1 millimolar. Buffer was added to bring the net volume of each well to 100 microliters. Fluorescence assays were performed in black NBS 96 well plates and read at a wavelength of 530/25 nm excitation and 590/35 nm emission using the Biotech Gen 5 microplate reader and imager software. Initial rates were calculated in RFU per minute, and curve fittings were processed using sigma plot to determine binding constants. So in conclusion, I1, I2, and I3 inhibited all MMPs tested with binding constants ranging from 7.5 to 82 micromolar. Although initially screened against MMP9 and having exhibited stronger docking scores with this enzyme, all three molecules had stronger inhibition against MMP1. L1 exhibited activating effects, also with stronger binding against MMP1 than MMP9. Differences in binding constants for a single molecule may be attributed to the structural differences among the MMP types. Further studies would benefit from understanding the type of structure that a promising inhibitor may possess in the search for MMP specific modulators to avoid negative side effects of broad spectrum MMPIs seen in failed clinical trials. The molecule's stronger action against MMP1 versus the other enzymes gives insight on a potential MMP specific structure that should be further explored. I just want to thank my mentor, Dr. Kumar.